biochemist Stephen Zamenhof experimented with brain development in chicken embryos, and he found that the brain stopped development at the same time as the glucose reserve in the egg from the hen was depleted. And so he added glucose, or amino acids that could be turned to glucose, to the egg. He waited the number of days that he knew the glucose would disappear, then punched a hole in the egg and injected the glucose, and showed that the injection of glucose allowed the brain cells to keep multiplying right up until hatching. And those chickens had a bigger brain than chickens had ever had and were more intelligent. And
Wow.
about the same time, Marion Diamond at the University of California was part of a group that was studying the effects of environment on the brain. And it was very Lamarckian. People didn't talk much about that, but just by giving rats an interesting environment instead of keeping them in a box, just giving them some playground toys basically and a bigger box and a few other rats to visit with, their brains got bigger and their enzyme activity changed. And the thing that least got talked about was that their offspring had bigger brains, bigger, more intelligent brains. And I don't know how many generations that went on, but I think I read that it was 4 generations they had seen a progressive increase in both the intelligence and the size of the brain. And during the same time, 1960s mostly, several other types of experiment and observation were made. Rats and dogs both had their brain development limited by excess estrogen, which lowered their blood sugar, made the glucose less available. The glucose was essential for brain development. So too much insulin or too much estrogen, which activates insulin, either of those would lower blood sugar and simply turn off brain cell multiplication. And polyunsaturated fatty acids were the other main thing that would stop brain development by interfering with the use of glucose. That at the same time was being called I forget the guy's name. Anyway, it was a well-recognized principle that fats block glucose oxidation. And so those 3 things were known to limit brain growth and intelligence. And but besides extra glucose simply being added, the level of progesterone was the main thing that protected against estrogen and the excess insulin effect. And to some extent, fatty acids would interfere with the polyunsaturated fatty acid blockage of glucose use. But progesterone affecting the hormone balance was found to produce this transgenerational advance of the brain, and it would show up as ovarian increased activity supporting increased brain size and intelligence. Having that in mind, I was looking around after I got my PhD, looking around for places where research could be continued. I found that the Catholic University in Valparaiso, Chile, had instituted, they were about to institute a new branch specifically to study the influence of nutrition on brain development. And I actually got a signed contract, never did from another university, but I was contracted to direct that program. And so I was planning to go to Chile, but then the 1973 Nixon-Kissinger coup stopped that whole project for 30 years or so.